Hello, this is Lawrence Bloomer at Morehouse College. This is a short video that I prepared to show you how to conduct the nucleotide blast of picked colony sequences. You should be following along using the handout Bean Beetle Microbiome Identification of Picked Colony 16S RNA DNA Sequence. The website we'll be going to is this site that's in the handout, the BLAST NCBI site. BLAST refers to Basic Local Alignment Search Tool, and this is a search engine that's available to us for free at the National Center for Biotechnology Information, which is part of the National Library of Medicine in the United States National Institutes of Health. Now, if we go we actually need to start with a sequence, a DNA sequence, and these should be the nucleotide sequences that were obtained from the sequencing of your picked colonies that you PCR amplified. Your instructor should have made available to you a PDF document of those sequences. Each sequence begins with a label. That label has a little arrow tool, and what you're going to want to do is highlight that entire sequence and then copy it, and then go to the NCBI site, pick nucleotide blast, and then the window at the top, you will paste that sequence into that window. So there it is, including the label. Now you'll notice there are these ends in the sequence at the beginning, and there are some at the end. There could be ends in the middle of the sequence. These are nucleotides that were not identified in the Sanger sequencing. But if they're at the very beginning of the sequence, and the majority of these are ends, it makes sense to just delete those. And so I'm just going to highlight those and hit delete to remove them. And then at the end of the sequence, highlight the two at the end and delete them. Then go to Choose Search Set. Make sure that this button is chosen, rRNA ITS databases, and 16S ribosomal RNA sequences for bacteria and archaea should be selected in this window. There are other selections, but you want it to be for bacteria and archaea. And then go and hit blast. Take a moment for this search to occur. There we are. I suggest you just show the top 10 sequences. And what we have are a series of matches that were made against your sequence, and the information provided here is the query coverage. The query coverage is how much of your sequence was incorporated in the match with this particular sequence. The E-value is the probability that the match occurred by chance alone. You want this value to be very small. You can't get smaller than 0.0, so that's fine. And the percentage identity is this value, and that's the number of nucleotides that are identical between your sequence and this sequence. Notice that all of these are Staphylococcus, but there are different species here. So these are very similar sequences with similar query coverage, the same E-values, and very similar percentages of identity. So it's very difficult to decide which species it is, but clearly they're all Staphylococcus, so the sequence that we searched must be from Staphylococcus. Your instructor may ask you to print this page and submit it. That can be done by simply going to the browser menu and choosing print. I suggest you just print the first page, this page. There is other information, so if you click on one of these sequences, you'll actually see what the sequence looks like. The query is your sequence, the subject is the sequence it matched it to. But the most important information for us is the information I showed you a moment ago. It's this search window showing the matches that were actually made. Now, once you've done this for every one of the sequences in your set, you'll want to enter that information in a spreadsheet. So, for example, here's a spreadsheet. The sequence that we searched a moment ago, in this case, the label here indicates it was from a Tuesday section from black-eyed pea. It was from beetle number 4. So I'm going to look for that. Here it is. And I could enter in here Staphylococcus. So your instructor may ask you to do this. It looks like I've got an extra A there. Your instructor may ask you to do this for each of the sequences that were submitted by your class and Sanger sequenced. You could fill in all of the different genera that were identified from the individual BLAST searches and then submit that data table. And certainly this particular data table could be an important part of your analysis of the research question that you were posing. If there are questions about the search that I've just described, certainly ask your instructor, or you can contact me at lawrence.bloomer@morehouse.edu. That ends this short video. Thank you.